Good morning, and I am Allan, and I'm one of the senior scientists here in the general lab. In this video, I'd like to show you an overview of G6PD screening tests. So basically, G6PD is a qualitative testing to test for the presence or absence of the enzyme G6PD, which is a very important enzyme in the red cell metabolism because it is mainly the effect of G6PD enzyme in red cell metabolism is it detoxifies all the toxic radicals that are being produced during cellular metabolism. Without that G6PD enzyme, we will not be having a defense mechanism to all the external or internal triggering factors that protect the red cells from hemolyzing. So G6PD, why we're doing this is because we want to screen a blood sample for the activity of the enzyme. It is not actually a direct test, but rather it is an indirect test to measure the G6PD enzyme. Why we're doing this is because we want to screen people who have inherited a disease that is X-linked by inheritance. So if since this is an X-linked inherited disease, majority of the affected individuals are mostly male, and female usually they have less severe manifestation of the disease. As you know that since it is X-linked, we know for a fact that it affects the X chromosome. Male have X and Y chromosome. Having said that, the X one is affected, they don't have extra X chromosome, as opposed to women where they have double X chromosome. So once one X chromosome is affected, they have an extra X chromosome that functions normally. So what we're going to do now is I'm going to show you how to screen for the presence or absence of G6PD enzyme. Okay, so let's get started. So what we have here is a Randox fluorescence immunospot test. That means we are going to combine, as you know, G6PD enzyme is located inside the cell, and so we need to liberate the enzyme outside of the red cell. So reagents that we are going to use is basically a buffer, okay? A buffer that contains the necessary cofactors and ingredients where the G6PD enzyme will act on, okay? Such as glucose-6-phosphate and the NAD-B. These are commercially prepared. So basically what it does is, the test does is that once we liberate the G6PD inside the red cell, for example, in this patient sample, we're going to take whole blood sample, take 5 microliters of the whole blood, and then put them in a 100 microliters of this buffer, or the working reagent that contains the buffer and a lysing agent known as saponin. Okay, so I'm going to show you how to execute the procedure. So let's start off by taking 100 microliters of
paper and label them as to your normal control, your partial deficient control, your deficient control, and your patient sample. So in this case, 6247. And then after 10 minutes, you can transfer, take 10 microliters in each, okay, from each of the following tubes, and then plot them into this filter paper corresponding to your labeled square, and then leave it for drying for about 10 minutes. And then once it dries up, then you can view this under a UV viewing light using ultraviolet light at a long wavelength. So since this is a fluorescence spot test, the presence of G6PD is actually an indirect method of detecting the enzyme by detecting its byproducts. Okay? So it is the byproduct of the reaction of the glucose 6-phosphate and not B. And then if there is G6PD in the red cell, it will act on these two compounds, and therefore it will reduce the NADP into NADPH, or nicotinamide adenine dinucleotide phosphate. Now this NADPH therefore is the one that we are going to measure, because this is the end product of the reducing property of G6PD, and it is the substance that gives fluorescence on your mixture. So the presence or absence of fluorescence is the indirect measure of the presence of G6PD enzyme in the patient sample. Okay, so this is how it looks like. It is completely dried up. So you have a normal control, partial deficient control, a deficient control, and you have 3 different patients in this example. So now this blotted mixture of your reagents and your patient whole blood will now be ready for viewing in a ultraviolet viewing box under long wave. So this is the source of the UV. This is a lamp. Okay. So it has 2 functions. One is for short wave. We're not going to use that. But long wave ultraviolet light is the one that we are going to use. So the difference between the two is just the wavelength of the ultraviolet light. So we are going to use the long wave ultraviolet light. And this is a completely dark environment inside, where you are going to place this filter paper inside, and then just press into the long wave, and then we can view it inside for the presence or absence of fluorescence. If you can see, okay, since we finished executing the procedural aspect of the G6PD, it is important, and I would say is one of the most important information that you need to know about performing G6PD is that you have to make sure that your sample has the following conditions. You should not have more than 22,000 WBCs, reticulocytosis, or recent blood transfusion, because they will give you false normal results. Why? Mainly because young red cells, white blood cells,
And recently, the working reagent. Again, the working reagent contains a saponin. A saponin is a substance that will lyse your red blood cells in order to liberate the G6PD enzyme inside the red cells. So, and then it also contains glucose 6-phosphogluconate and NADP. So these are the necessary cofactors where the enzyme will act on later on. Okay? Then we are going to get 5 microliters of the whole blood. So there you go. So it is important that for your patient tube, okay? So remember, you have to identify each tube so that you will not be confused. So I am putting the 5 microliters of the whole blood into my tube labeled patient 6247. Okay, mix well. And then you can continue with your controls, or either you start with your controls, or either way, so long as you can identify. So these controls are commercially prepared, and we just treat them as patient sample. Okay, so we have the normal control, the partial deficient control, and the deficient control, which we prepare in-house. Okay, so by collecting, okay, so this is my normal control. Remember, 5 microliters, exactly the same as the patient. So normal, will place it inside a tube labeled N for normal, mix well, and you can start, continue with your deficient, partial deficient. Again, 5 microliters of the partial deficient control placed into a tube labeled partial deficient. Mix well. And then finally, with our deficient control. Deficient control is actually an in-house, prepared in-house, where we collect whole blood samples from our patient, and then we subject the whole blood into 50 degrees Celsius water bath in order to deactivate the enzyme. So once you deactivate the enzyme by heat, you are basically destroying the enzyme, and it will act as your deficient control. Okay, so this is now our deficient control. Okay, so now we're done with preparation. We just allow the solution, the mixture, for 10 minutes. Okay, and then after 10 minutes, it is now ready to blot into a Guthrie filter paper. So while waiting for the incubation at room temperature of your mixture, you can label your
blotting filter paper and label them the buffer and a lysing agent known as saponin. Okay, so I'm going to show you how to execute the procedure. So let's start off by taking 100 microliters of the buffer that contains the G6PD working cofactors. So basically, what actually happens inside the red cell is we are trying to create an in vitro environment, okay, where the G6PD enzyme act on exactly the same as what happens inside the red cell. So this particular solution contains glucose-6-phosphate and NADP. Okay, so it is commercially prepared. So these are the necessary cofactors for the red cells for the glucose-6-phosphate dehydrogenase enzyme in case there is enough activity or presence or absence of the G6PD enzyme in the sample. Okay. So what I'm doing now is I am putting 100 microliters of this solution into the corresponding test tubes labeled, okay, as your deficient control, your partial deficient control, and your deficient control. And also we are going to create another extra tube for your patient. So in case you have more than one sample, then you add more tubes along with your 3 levels of control. Okay? It is important for you to place all your controls and your agents at room temperature for at least 10 minutes. Reach at room temperature to be able to make sure that the reagents will be at its optimum reactivity. Okay, so moving on, do not forget that labeling is very, very important in any specimen processing. Okay, so you label your tubes as to your deficient control, partial deficient control, and your normal control. Okay. So depending on each lab, they have their own protocol in labeling. So long as you can identify your tube and your controls, it is fine. Okay. So now, since all our samples are barcoded, we are trying to get the last 4 digits of the barcode number to label your patient sample. In this case, we have 6247 for this particular patient. So 6247. Okay? So let's continue. We will continue adding 100 microliters of the working reagent. Again, the working reagent contains a saponin, A saponin is a substance that will lyse.
Okay. Since we finished executing the procedural aspect of the G6PD, it is important, and I would say is one of the most important information that you need to know about performing G6PD is that you have to make sure that your sample has the following conditions. You should not have more than 22,000 WBCs, reticulocytosis, or recent blood transfusion, because they will give you false normal results. Why? Mainly because young red cells, white blood cells, and recently transfused blood, they normally have G6PD enzymes. So if your sample has that conditions, you will end up having a false normal G6PD enzyme activity. So that's it.